This is part 2 of our lecture series on urinary system. We'll begin by discussing tubular secretion. On the last lecture, we discussed the process of glomerular filtration and tubular reabsorption. Glomerular filtration is always a passive process. Okay, that process never requires energy. Tubular reabsorption may be passive or active, depending on what's being transported. Tubular secretion, however, is always an active process. It always requires energy. Okay? And that's because it always requires moving substance against their concentration gradient. And the purpose of tubular secretion is to eliminate excess or undesirable substances. And that includes some drugs, metabolites that are bound to plasma proteins. And since plasma proteins are too large to be filtered through the glomerulus, so anything that's bound to them would have to be eliminated through tubular secretion. Other things that are secreted are undesirable substances that were initially passively reabsorbed, like urea, uric acid. Also, if you have too much potassium in your body, this is something we'll discuss in conjunction with the renin, angiotensin, aldosterone system later in this lecture. So excess potassium can also be eliminated through the kidney. And since your blood has to be maintained with a certain acidity and alkalinity, the long-term regulation of blood pH can be regulated through altering the amount of hydrogen. Moving on to talking about excretion. We've so far discussed glomerular filtration, tubular reabsorption, and tubular secretion. So it should make sense that for any substance that is filtered by our kidney, if you subtract the amount that's being reabsorbed and then adding the amount that's being secreted, we will eventually get the amount that will eventually be excreted from our body. Okay, and this is a value that can be easily measured by doing analysis of our urine. However, just by knowing the rate or the amount that of a substance that our body is eliminating tells us nothing about how our kidneys are handling that substance. Okay, so in other words, it tells us nothing about the details of how well our kidneys are functioning. And the renal handling of a substance and glomerular filtration rate, GFR, is often of high clinical interest. For example, glomerular filtration rate is routinely used as an indicator of the overall renal function. Our kidneys are not easily accessible and our nephrons are microscopic. Okay, and so for these reasons, physicians and scientists have to have ways of assessing renal function using only urinalysis or samples of urine and samples of blood, okay, without doing any more invasive procedures. Okay, and so for them to do this, we first have to understand a concept called renal clearance. Renal clearance is an abstract concept that describes how our kidneys handle substance based only on blood and urine analysis. So in other words, it's a non-invasive way of looking at kidney function. For any solute that is cleared only by renal excretion, we can express clearance in the form of volume of plasma passing through the kidney that has been totally cleared of that solute within a given amount of time. And because this is an indirect way of thinking of excretion, this is a concept that often is very challenging for a student to grasp initially. So notice that, again, it's expressed in terms of volume of plasma per unit time. It's not expressed in terms of the amount of substance per unit time. Through scientific experiments, scientists have found a substance, which is a plant extract called inulin. And when it's injected into blood, okay, it's freely filtered into Bowman's capsule, but it's neither secreted nor reabsorbed by the renal tubules. In other words, 100% of the inulin that is filtered into the tubule, it will be excreted. And so for this reason, the clearance of inulin is used or can be used to calculate GFR. Okay. And so if we compare the clearance rate of substance with the clearance of inulin, we can get an idea as to how our kidneys are overall handling that substance. So if you look at the clearance of a substance and the clearance is less than the clearance rate of inulin, then that tells us there's a net tubular reabsorption of that substance. Whereas a clearance higher than the clearance of inulin would indicate there's net tubular secretion. Okay, the key word is net. Okay, in the case of net tubular reabsorption, okay, it doesn't mean that there's no tubular secretion at all. It just means that there is overall more tubular reabsorption than secretion. Okay, and likewise for the net tubular secretion. Now, when the clearance of that substance is 0, that tells us there's 0 amount of plasma being cleared of that substance within that given amount of time. In other words, this indicates that the substance is completely reabsorbed. Here we're going to look at the renal clearance examples for a few other substances. So we've already discussed inulin. It's freely filtered and neither secreted nor reabsorbed. So that means 100% of what is freely filtered into the Bowman's capsule ends up being excreted. Okay, and so therefore the clearance of inulin equals GFR. Glucose clearance is very different. Our bodies are really good at retaining glucose. Okay, and by the end of proximal convoluted tubule, usually 100% of the glucose is reabsorbed. So glucose is small enough to be freely filtered. So it's freely filtered into a Bowman's capsule, but by the end of proximal convoluted tubule, usually 100% of it has been reabsorbed. In other words, the glucose clearance is 0 milliliters of plasma per minute. In other words, none of the plasma could be cleared of glucose in that given amount of time. The clearance of urea is an example of net reabsorption. We see that urea is another molecule that's freely filtered into Bowman's capsule. However, unlike inulin, where none of it is reabsorbed, in the case of urea, some urea ends up being reabsorbed, and so not all of it that's freely filtered will end up being excreted. And so if we compare the clearance of urea, we see that it's less than the clearance for inulin. And that indicates that there is net reabsorption of the substance. In the case of clearance of penicillin, which is an antibiotic that you all have heard of, this is an example of net secretion. Okay, so it's freely filtered into renal tubule or into Bowman's capsule and the renal tubule. But there is going to be some additional penicillin that ends up being secreted. In other words, it's going to go from the circulatory system and then into the tubular lumen. Okay, so that means the amount that ends up being excreted is more than what is being filtered here. Okay, and so if we look at the clearance of penicillin, we see that this is a higher value than the clearance value for inulin. In other words, there is net secretion. So why is all this important? Well, this is important because it shows how we can measure GFR in a person by taking only their blood and urine samples without any invasive procedures. Inulin clearance can be used in science research to estimate GFR. However, it's not practical for routine clinical applications because first of all, it doesn't occur naturally in the body. And secondly, it has to be administered continuously through intravenous infusion. Okay. And so for this reason, inulin use for estimate of GFR is restricted to science research only. In clinical settings, physicians will use creatinine instead to estimate GFR. So creatinine is a waste product that comes from normal muscle use and from meat protein in your diet. And in a healthy kidney, it will remove creatinine from the blood. Okay, and then the creatinine then passes out your body through the urine. Unlike inulin, creatinine is constantly produced by the body and does not need to be administered. Okay. So normally, the production and breakdown rates of creatinine is relatively constant, and the plasma concentration of creatinine doesn't vary much. Even though creatinine is always present in the plasma and is easy to measure, it doesn't provide a super accurate estimate of GFR, because there is a small amount that's actually secreted. Okay. In other words, the amount that is excreted will end up actually being slightly higher than what's being filtered. But because the amount that is secreted is small enough, and because our body's production rate for creatinine is relatively constant, so in most cases, in most people, creatinine clearance can and is an acceptable use for GFR estimate. Okay, and is what is used routinely clinically to estimate GFR, and in other words, indirect measure of renal function. Let's talk about the fluid compartments of our body. So the amount of water in your body takes up around 60% of your body's weight. In other words, 60% of your body weight comes from water. Okay. And if we take a look at the different compartments in our body where the body exists, we can divide it into intracellular fluid and extracellular fluid. Intracellular, as the name indicates, is all the fluid volume located inside the cells. So if we're to measure the volume of water in each cell and then add it up across all the cells in our body, okay, that occupies about 2/3 of our total body water. Okay, whereas 1/3 of it lies in extracellular fluid. Okay, and that extracellular fluid is fluid in the interstitial space, in other words, in between the cells, or I should say, and the fluid inside your blood vessels. So in other words, plasma, or the intravascular fluid. Okay, and if we look at the amount of ECF found in our intravascular space versus in the interstitial space, we see that around 1/5 of the ECF volume is located inside our blood vessels, whereas 4/5 of it is in the interstitial space. Now, looking at the chemical composition of these different fluid compartments, we see that the chemical composition of plasma and interstitial fluid are very similar. And as we previously learned, between our endothelial cells, we have those intercellular clefts. Those holes allow most of those small ions, as well as water, to freely cross in between the plasma and the interstitial space. Okay, and I explained why those two fluids have very similar composition. Okay, and these spaces are typically high in sodium, chloride, okay, bicarbonate ion, okay, very little potassium. Okay. Do notice one difference in between these two spaces. Plasma typically has more protein and ions than your interstitial fluid. And these, because of their large size, they're not as easily, they can't easily cross the endothelial cells into the interstitial space. So under normal circumstances, most of them will stay inside this intravascular compartment. Now, taking a look at the intracellular fluid, the ICF, we see that the ion composition is very different from what we see in the extracellular compartments. We see a high concentration of potassium and phosphate. There is also a high concentration of these anionic proteins. One of the most important goals of homeostasis is to regulate fluid volume of our body. Each day, the water intake of our body comes from metabolism, food, and beverages. And of course, you can drink more or less, so this amount can be highly variable. And the amount that your body outputs has to be balanced. Otherwise, if you excrete too much, if you lose too much, you eventually be dehydrated. Okay? And the output that our body produces is in the form of feces, sweat, insensible loss through skin, like evaporation, through lungs, through respiration. Okay, and finally, the greatest amount of fluid loss each day usually comes from urine. Okay, and this amount can actually be heavily modulated depending on using our kidneys function. So now we're going to look at how our kidneys are able to regulate water balance. This ties to an important concept called countercurrent mechanism system. So I've included some videos here that are sort of long to watch here, but please take a moment to pause our video and watch these videos on your own. So ever wonder why your kidneys are worth so much on the black market? Well, your kidneys filter as much as about 180 liters of blood every single day. Recall that our body has only, on average, about 5 liters of blood. So that means our kidneys filter 30 to 40 times the amount of blood that we have each day. Okay, what's even more amazing is that of that 180 liters of fluid that enters into our kidney nephrons, only 1.5 liters on average end up being excreted as your urine. That is less than 1% of what's being filtered. So where do you think that remaining 99% filtered volume went? That's right, it actually was reabsorbed back into our circulation. Okay, and just how is our kidney able to accomplish this phenomenal task of conserving water? And they do this through the countercurrent mechanism, which we'll now discuss. Well, so what is this countercurrent mechanism? The countercurrent mechanism is so named as it involves fluid flow in opposite directions through adjacent segments of the stem tube connected by a hairpin turn. Okay. And as you'll see, this setup allows for exchange of substances between two adjacent segments. And this applies to loop of Henle as well as vasa recta. Now, the purpose of the countercurrent system is to set up a concentration gradient across the medullary interstitium that can later be used by the distal convoluted tubule and the collecting duct to modulate the amount of water reabsorption under influence of aldosterone and ADH, which we'll discuss later in this class. And the countercurrent system relies heavily on the medullary interstitium, having a high osmolarity. I'm going to say that again. The countercurrent system relies heavily on this medullary interstitial fluid having a high osmolarity. Okay. In other words, this interstitial space, this medullary interstitial space has to be very salty in order for this system, for this mechanism to work. And this high osmolarity in the medullary interstitium is what drives the osmotic movement of water out of the renal tubules, later in the distal convoluted tubule and in the collecting duct. Now, there are a few key players that interact with the medullary osmotic gradient. First, we had the loop of Henle, which acts as the countercurrent multiplier because it helps generate the high osmotic gradient within the medullary interstitium. Then the vasa recta acts as the countercurrent exchanger in helping to maintain the concentration gradient that's being established in the renal tubules and in the medullary interstitium by the long loop of Henle. The collecting duct of our nephrons can then use that osmotic gradient in the medullary interstitium to then adjust the urine's osmolality. So to summarize, the long loop of Henle of the juxtamedullary nephrons and vasa recta will work together to establish and to maintain an osmotic gradient within the renal medullary interstitium. In other words, these two structures work to set up and to maintain that salty environment within the medullary interstitium. And then the collecting duct then utilize that gradient to adjust the urine to its final volume and final osmolality. We just said that the long loop of Henle of these juxtamedullary nephrons act as countercurrent multiplier to establish the osmotic gradient. There are three properties that allow it to establish this osmotic gradient. First, the countercurrent flow filtrate through the adjacent parallel sections of the loop of Henle. This is the countercurrent system that we talked about. And then secondly, the second property is that the descending limb loop of Henle is permeable to water, but not to salt or solute. Okay, so there is passive water reabsorption that happens here as the filtrate is flowing through the descending limb. The third property of the loop of Henle being that the ascending limb is permeable to salt and solute, but not to water. Okay. So in this segment of the loop of Henle, there is active reabsorption of sodium and chloride. Okay, and these properties establish a positive feedback cycle that uses the fluid flow to multiply the gradient. In other words, the more sodium chloride the ascending limb can extrude out of the lumen, the more water can diffuse out of the descending limb, and therefore the saltier the filtrate in the descending limb will then become. So the ascending limb then uses this increasingly salty filtrate that's left behind in the descending limb to raise the osmolality of the medullary interstitial fluid even further. Okay. And so this establishes a positive feedback cycle that produces the progressively higher osmolality of the fluid in the descending limb and in the interstitial fluid. So once again, in the ascending limb, salt is actually pumped out. And so we see that the filtrate becomes more and more dilute as it ascends. Okay. And so this establishes a concentration gradient within the medullary interstitium, okay, which then allows for the passive diffusion of water out of the descending limb over here. Okay. And that increases the osmolarity of the filtrate entering into the descending limb. Okay. And that gradually, progressively saltier filtrate that makes it easier for more salt to be then extruded in the ascending limb again. So this is a positive feedback cycle. Because this is a difficult concept initially for students to understand, I'm going to explain this again. As water and solutes are reabsorbed, the loop first will concentrate the filtrate and then later dilute it. And so this positive feedback cycle with the initial concentration followed by dilution, this sets up an environment of countercurrent exchange. In other words, there is an increasing sodium chloride level as we delve deeper down into the renal medulla. And this makes it easier for water reabsorption to later occur in the descending limb loop of Henle and also later in the collecting duct. Now, countercurrent multiplication is an active process. Okay, it establishes the osmotic gradient in the medullary interstitium. The luminous surface of the tubular cells in the ascending limb loop of Henle contain these special cotransporters called NKCC cotransporter, and its name indicates that it transports 1 sodium, 1 potassium, and 2 chloride ions out of the tubular filtrate. And this NKCC cotransporter is an example of secondary active transport process because it harnesses the potential energy stored in the sodium gradient established by the sodium potassium ATPase to move these ions out of the tubule. So what role does the vasa recta play in all of this then? Well, recall that a descending limb loop of Henle is permeable to water, but not to salt. If we didn't have a mechanism to remove this water out of the medullary interstitium, then all that water will eventually undo all the osmotic gradient that your loop of Henle had worked so hard to establish. The vasa recta is highly permeable to water and to solutes or salts. And the countercurrent flow of fluid will move through 2 adjacent parallel sections of the vasa recta, similar to what we see in renal tubules in a loop of Henle, but except that now those segments are as permeable to salt as it is to water. And the countercurrent exchange occurs between each segment of the vasa recta with its surrounding fluid. As a result, we see that the blood within the vasa recta remains nearly isoosmotic to the surrounding fluid. And the vasa recta are able to reabsorb water and solute into the general circulation without undoing the osmotic gradient that's being established by our loop of Henle, or in other words, established by our countercurrent multiplier. And so the end result of this arrangement is that the blood flowing through vasa recta removes the water reabsorbed from the loop of Henle. And so without the vasa recta, water moving out of the descending limb loop of Henle would eventually dilute the medullary interstitium. And so therefore, the vasa recta plays a very important part in keeping the medullary solute concentration high. So this process called countercurrent exchange, and vasa recta being called the countercurrent exchanger, This process is a passive process, unlike the process of countercurrent multiplication. Okay, so countercurrent exchange is passive process. It maintains the osmotic gradient in the medullary interstitium. And so this is an image that summarizes the concepts that we just discussed. We've got the loop of Henle, where the extending limb contains NKCC cotransporter that actively pump out sodium, chloride, and potassium out of the tubular lumen. And that allows us to establish a salty environment in that medullary interstitium, which is then used to passively reabsorb water through osmosis. Okay. And to prevent that water being reabsorbed from diluting the medullary interstitium, we have vasa recta acting as the countercurrent exchanger to pick up that water. Okay, so that water isn't staying around in the medullary interstitium, diluting the medullary interstitium. Okay, so blood in the vasa recta removes water, leaving the loop of Henle. In the distal convoluted tubule and in a collecting duct, there is variable secretion and reabsorption of solutes and water. These regions are under hormonal control, specifically under the control of a hormone called aldosterone and another hormone called ADH, antidiuretic hormone. Here, let's take a look at what happens under the influence of ADH. ADH, antidiuretic hormone, also known by the name of vasopressin. ADH, if you recall from earlier this semester, ADH is a hormone secreted by our posterior pituitary. Okay, it's actually synthesized by hypothalamus, but secreted by our posterior pituitary. It's released into blood, and when it gets to our kidney, okay, specifically in the collecting duct, it triggers the insertion of aquaporin channels, or these water channels, into the luminal surface of the tubular cells in collecting duct, and also in some distal convoluted tubule. And this leads to more water being reabsorbed. So what happens to the filtrate, or the concentration of filtrate, when you extract more water out of it? Correct, you will concentrate it, you'll make it more concentrated. But depending on the level of ADH secretion, more or less water channels or aquaporin channels can be inserted, and therefore more and less water can be conserved by our kidneys, depending on the hydration status of our body. So putting it all together, our kidneys are able to conserve water, but in order for it to move water, it has to first move sodium. The way our kidneys are able to accomplish this is through a mechanism called the countercurrent system. So that encompasses countercurrent multiplication and countercurrent exchange. And the purpose of the countercurrent system is to set up a concentration gradient across the medullary interstitium that can then be utilized by the distal convoluted tubule and by the collecting duct to modulate the amount of water reabsorption that can occur under the influence of the hormones called aldosterone and ADH. So later in this lecture, we'll take a closer look at aldosterone as well as ADH. We're moving on to talking about sodium balance in our body and how that allows for regulation of ECF volume. As we've already seen, water reabsorption in our kidneys is closely tied to the reabsorption of solutes. And the reabsorption of the solutes rely on the sodium gradient across the luminal membrane of those tubular cells. So for this reason, the total amount of sodium in the body is the primary determinant of ECF volume. And this ECF volume is going to directly impact our blood pressure, because that intravascular space containing our blood is part of that extracellular space. So which hormone is the most important in regulating renal sodium reabsorption? The answer is aldosterone. You may recall from the endocrine system chapter that aldosterone is a steroid hormone synthesized in the adrenal cortex. And its trigger is hypotension, a drop in blood pressure. Alternatively, it's also triggered by a rise in plasma concentration of potassium. So when you experience a drop in blood pressure, it activates a complex cascade of events called the renin-angiotensin-aldosterone system, which we'll talk about soon. This pathway eventually stimulates the adrenal cortex to release aldosterone. Aldosterone, like all hormones, travels through blood. But when it gets to kidney, it will act on the principal cells or P cells of collecting duct, causing these cells to increase sodium reabsorption along with increased potassium secretion. Okay, so it allows your body to regain more sodium and dump more potassium. And recognize that the results are the opposite of the trigger, right? We've got hyperkalemia triggering this pathway and ending with increased dumping of potassium. Okay, and whereas the primary trigger of hypotension, right, results in increased sodium reabsorption. And remember, what follows sodium reabsorption is water. Right? Water gets passively reabsorbed by osmosis following this sodium reabsorption. So that means at the end of this pathway, we're also going to bring our blood pressure back up. Okay, so to summarize, the more aldosterone secreted, the more sodium reabsorption, okay? And the more potassium is wasted, meaning it's secreted and lost from the body. Here we've got a video on renin-angiotensin-aldosterone system, sometimes referred to as just the renin-angiotensin system. So take a look at this video and before we move on to the next slide. So how do we regulate the release of aldosterone from adrenal cortex? Well, the answer lies in this hormone called angiotensin II, as well as a complex pathway called the renin-angiotensin-aldosterone system, RAAS. Okay, and this is a complex series of events that is extremely crucial in the maintenance of our blood pressure. This pathway begins when the juxtaglomerular granular cells in the afferent arteriole of our nephron secrete an enzyme called renin. And recall from previous lecture that renin gets secreted when there is decreased renal perfusion. Okay, and renin, upon its release, it converts an inactive plasma protein called angiotensinogen into angiotensin I, and angiotensinogen is secreted by the liver. Now, angiotensin I can then be converted to angiotensin II by this enzyme called the angiotensin converting enzyme, ACE. Now, this conversion by ACE can be found to occur in the lungs, but also as well as the endothelial cells of blood vessels throughout our body. Now, when this angiotensin II in the blood reaches the adrenal gland, it causes the synthesis and release of aldosterone. Okay, so from this point on, this takes a course that we just discussed in the last slide, which is aldosterone then causes increased sodium reabsorption along with increased potassium secretion. Okay, but also notice that angiotensin II has a number of other effects. It increases sympathetic activity, okay, and it also increases arteriolar vasoconstriction, and this in turn helps achieve that resultant increase in blood pressure. And furthermore, angiotensin II acts to stimulate the pituitary gland, specifically the posterior pituitary, to increase its secretion of ADH. And ADH, as we previously mentioned, allows for increased water reabsorption. Okay, and there's an important distinction between the effect of ADH and aldosterone. Okay, and something we'll re-emphasize again in 2 more slides. But notice that ADH causes free water reabsorption without being accompanied by sodium reabsorption. Okay, whereas the effect of aldosterone is that it causes sodium reabsorption followed by water reabsorption. So both sodium as well as water, instead of just water alone here. In any case, all of these series of events result in water and salt retention, and this effectively increases the circulating volume. Okay, so there's more volume in your blood vessels, and that means increased blood pressure. Okay, and this will then act to feedback to negatively inhibit or produce a negative feedback back at the starting point on renin secretion. We're now going to do a side-by-side comparison of aldosterone and ADH. Aldosterone, of course, is secreted by the adrenal cortex, whereas ADH is secreted by the posterior pituitary. And notice there are a slight difference in the triggers. Aldosterone is triggered primarily by decrease in body salt content or body's sodium content, which triggers the renin release, increasing angiotensin II. So this is the renin-angiotensin pathway we just discussed. Okay. Alternatively, it's also triggered to release by hyperkalemia. In other words, rise in potassium level in our blood. So aldosterone, when released, it acts on the distal convoluted tubule and on the collecting duct. Okay, and resulting in increased sodium reabsorption along with increased potassium secretion. And that allows us to restore our plasma volume, okay, because the water then follows by osmosis. In other words, the overall effect is an increase in ECF volume without dilution, because the water that's reabsorbed is accompanied by the salt. Or we should say the salt that's reabsorbed is accompanied by the water. However, if there is too much aldosterone secretion, you will expect to see a drop in the person's plasma potassium. Okay, because that's sort of a side effect of having aldosterone secretion. Now, ADH is triggered to release by increase in ECF osmolality. Okay, and increase in ECF osmolality can happen when, say, you're dehydrated or you've ingested too much salt. Okay, and a rise in sodium concentration in plasma also triggers ADH release. Okay, and ADH, once released, acts on the collecting duct of our kidney to result in increased free water reabsorption. I say free because it's not accompanied by sodium. Okay, and that allows for increased plasma volume with dilution. Okay, meaning there's a drop in ECF osmolality. Here's a great video on ADH. Please take a look at this before moving on. And we've also got a video comparing aldosterone and ADH. Take a look at these as well.